Okay, in this chapter 5, we are going to learn about metabolisms and enzymes. So there are three subtopics within this chapter. The first one, we are going just a little bit about metabolism, and then the longest, which is enzymes, and then the third, application of enzymes in daily life. So let's start with metabolism. Okay, metabolism. Metabolism, in a simple ways, it means the summation of all reactions that occur in our body. In the sentence, we say that metabolism refers to all chemical reactions that occur in a living organism. In the living organism, because animals and fungi also have metabolisms in their body. And then there are two types of metabolism, which is the first one, catabolism. Secondly is the anabolism. What is the meaning of catabolism? It is the process of breaking down, the breakdown of complex substance into simple substance. In the equation, we written as followed. A is the substrate undergo breakdown process, becoming products, which is B and C. And this arrow shown the breakdown process that happen in the reactions, in these chemical reactions. And then this reaction usually releases energy. And the example of this reaction that release energy, the breakdown of complex into simple substances, is the glucose. Breakdown of glucose during cellular respiration to generate energy. How to remember that this glucose, this example will show that catabolism is releases energy. So there are one equation that we already learned before during primary school, which is glucose react with the oxygen will become carbon dioxide and water, and this reaction will release energy. We know that glucose is monosaccharide and the simple sugar, but in this case it is complex substances because it has 6 carbon, and 6 carbon is bonded by chemical bonding, and this is considered as complex, and energy will be released in order to release the carbon dioxide and water, which is the byproducts. Okay, so let's look at the anabolism. Anabolism is the reverse of the catabolism, which is process of synthesizing complex molecules from simple molecules. In this part, it uses the term molecules. And then okay, when as we write complex molecules into simple molecules. And then the equation is reversed as catabolism. A plus B, the substrate will combine synthesizing C, which is the product. And then this reaction will absorb energy. And then this reaction as an example, which is anabolic reactions, is the formation of glucose during photosynthesis. Formation of glucose during photosynthesis. Glucose, which is C6H12O6. Okay. That's all about the metabolisms. Enzyme. What actually enzyme is? It is an organic catalyst that made up of proteins produced by living cell organisms. An organic catalyst that is mostly made up of proteins and is produced by living cell organism. This enzyme can speed up the chemical reactions, help speed up reactions. So because of that, it is important in daily life for having an enzyme, because this enzyme, without it, thousands of chemical reactions that are necessary to sustain life would simply be too slow. So it is enhance the reactions so it can take place very fast. And then there is a formation of an enzyme substrate complex. So here, the reactants of enzyme catalyst reactions are known as substrate. And then this substrate will bind to specific location that is known as active site and forming an enzyme substrate complex. Enzyme nomenclature. This is how we naming the enzymes. It is based on the substrate or reaction it catalyzes. For example, sucrase, lactase, and maltase. These enzymes are responsible for break down sucrose, lactose, and maltose into their simplest units. And then the name of enzyme is derived by adding ASE to the name of the substrate it catalyzes. For example, lactose, we add with water. By using the enzyme lactose, will produce glucose and galactose. General characteristics of enzymes. So there are how many characteristics? Characteristic that we should know is 1, 2, 3, 4, 5, 6, 7, and 8. So there are 8 characteristics. The first one is enzymes are required in small quantities and are reusable, meaning just a small amount can be used, and can be used repeatedly. Secondly, the structure of enzymes remain unchanged and are not destroyed after a reactions. So they are not changed or damaged by the reactions that they catalyzed. And then the reactions of enzymes are specific. These tell us that the enzymes are very specific. One enzyme responsible to break down the specific substrate. Most of the reaction catalyzes by enzymes are reversible, meaning that if we already break down the substrate into its products, we cannot going back from the product to the substrate. We can by using another procedure, and then can be slowed down or stopped by enzyme inhibitors. So there is situation where inhibitors and substrate are the same shape and they are compete with each other to get the enzyme. So if the inhibitors uh, get into the enzyme, thus the substrate cannot undergo breakdown process. And then some enzyme need cofactors to work more efficiently. Usually, cofactors are non-protein substance. They are help with the reactions. This is usually like ions, for example, calcium ions, zinc ions. They are very helpful. For example, in our mouth, there is salivary amylase. This salivary amylase, with the presence of chloride ions, it can increase the chemical reaction salivary amylase activity in our mouth. It can obviously speed up biochemical reaction. And lastly, act rapidly. Enzymes. There are two types of enzymes, which is intracellular enzyme and extracellular enzymes. Intracellular enzymes is the enzyme that are synthesized in a cell for its own use. For example, hexokinase enzyme. This is enzyme that used to convert glucose into ATP during the respiration process. And then extracellular enzymes. These enzymes secreted outside of the cell, which is trypsin enzyme. This is enzyme that found in the lumen of the small intestine that can digest protein. Production of extracellular enzymes. Usually, extracellular enzyme is the enzymes that are produced within the cell and then secreted outside of the cells in order it to be functional. And the process, it has 6 steps. The first one, here they are ribosome, and we need to remember that ribosome will produce protein. Ribosome produce protein. And the second point, this protein will go to the rough endoplasmic reticulum through the lumen. And then the third point, as the protein was inside of the rough endoplasmic reticulum, will be form a bud and will pack into transport vesicle. And then the fourth point, this transport vesicle will be carry the protein into Golgi apparatus. And the fifth point, as the protein go into the Golgi apparatus, the protein is modified into enzyme. Protein will modify into enzyme. And this enzyme will pack into vesicle. And this time we call it as secretory vesicle. And the sixth point, the protein will secreted outside of the cell. So we can say that secretory vesicle fuse with the plasma membrane the enzyme will be secreted outside of the cell. So let's we make a fully sentence. The first one, the protein is synthesized by the ribosome. Ribosome is the site of protein synthesis. And the second point, proteins will enter the rough endoplasmic reticulum through the lumen. So the sentence will be like proteins synthesized by the ribosome enter the lumen of the rough endoplasmic reticulum and are transported through it. The points, proteins will be packed into transport vesicle. Once it reaches the end of the rough reticulum, the membrane buds off to form transport vesicle. Okay, the fourth point, the protein will that has transported into transport vesicle go through the Golgi apparatus. So the center will be like the transport vesicle. Moves toward the Golgi apparatus and then fuses with it. The fifth point. Since we know the fifth point is where when this enzyme has been modified, it must be transported outside of the cell, so we can write as the Golgi apparatus the proteins are modified into enzymes and secreted in secretory vesicles that are formed from the tip of the Golgi apparatus. And lastly, this secretory vesicle will transport the enzyme within it and then fuse with the plasma membrane so that it can secrete the enzymes out of the cell. Secretory vesicle towards the plasma membrane. Extracellular enzymes. Okay, so let's recap. That's a too lot. Okay, the first one is the ribosome will synthesize protein. Second point, the proteins will enter the rough endoplasmic reticulum through the lumen. And the third point, the proteins will be packed into transport vesicle. The fourth point, the transport vesicle that contains proteins will transport the proteins towards the Golgi apparatus and fuse with it. And then the fifth point, the Golgi apparatus can do its function to modify proteins into enzymes, and then it can pack it into secretory vesicles in order to secrete it outside of the cell. This secretory vesicle that have been formed that has enzyme within it will secrete it to plasma membrane. We secrete it outside of the cell by fusing it with plasma membrane. So that's all about it. Activation energy. So what is actually activation energy is? For most reaction that occurs, for example, anabolic and catabolic reactions, this chemical reaction must gains an energy. This collision energy is required and must be surpassed the energy barrier so that the chemical reaction can get started. For example, this purple line, the activation energy without enzyme, and this red line have enzyme in the reactions. So it is energy needed to push molecules to a higher energy level. Activation energy is energy needed to break the bond in the substrate before reaction can occur. In the presence of enzyme, it can lower the activation energy thus making the rate of biochemical reaction increases. The mechanism of enzyme action and factor changes. There are many factors that can affect the action of the enzyme. These factors can result in the chemical changes of the reactions. These factors due to physical conditions, for example, the temperature, and second is the pH. The third, effect of substrate concentration. And the fourth is the enzyme concentration. So factor number one, which is the temperature. First of all, this is the graph of the rate reaction against the temperature. And this graph will show us at below 0 to 10, the temperature is very low. The temperature is too low. The rate of reaction catalyzed by the enzyme is also slow. Why it is slow? Because the enzyme is still not activated and not function at that time. And then as the temperature rises, the kinetic energy of the substrate molecule and enzyme also increase. This will lead to the increase of the frequency of effective collision. Between the substrate molecule and enzyme molecule. Because of these three factors, 1, 2, and 3, they will lead to the increase of the rate of reaction. So another important point is with every rise of 10 units of temperature, the rate of reaction also double until it reaches the optimal temperature. This optimal temperature is 37 degrees Celsius. For example, like 0 to 10, it is A, and then 10 units of temperature increases to A, and 10 units increase in the temperature for air, for A, and so on. So with the rise of temperature of 10 degrees Celsius, rate of reaction will be double until reach the optimal temperature. So again, the point number 1, during 0 to 10, the temperature is low and the reaction rate of reaction is also low because the enzyme is still not function or not activated. And then the temperature increase. As the temperature increase within the slope, the kinetic energy also increases, since the energy supply in a heat form able to the reactions the system to absorb, and this can lead to the increase of the frequency of effective collisions between the substrate and the enzyme molecules. So because of these increases, the rate of reaction also increase. So another important point is when as the increases of temperature in 10 units, the rate of reaction also will be double until it reach the optimal temperature, which is 37 degrees Celsius. It is our own body temperature. Okay, point number 2, which is the at the optimum temperature. The point is the enzyme reaction is at maximum. And we can also mention that optimal temperature is 37 degrees Celsius. The third point we need to explain after the 37 degrees Celsius. The third point, after reaching the optimal temperature, the enzyme activity stop at 60 degrees Celsius. Why it is stopped? Because the enzyme is denatured. And what happened during the denaturation process? The substrate does not complement. The active site of enzymes. Another factor that can affect the enzyme activity is the pH. So enzyme is very sensitive to pH. And here we have a graph showing the rate of reaction against the pH. So basically we have three enzymes, main enzymes that we need to know, which is the pepsin. The pepsin can be found inside our stomach to digest the protein, and then we amylase, which is can be found in the mouth, and then trypsin. Trypsin can be found in the small intestine inside of the ileum. Let me know that first of all about pepsin. Pepsin is very acidic, and it is can be found in the range of 1.5 to 2.5. This pH can make sure the pepsin can function effectively and optimum at that pH. And then amylase is inside our mouth, and our mouth basically are neutral, which is at the pH 7, and works at the 6.8 pH of 6.8. And then the trypsin is the most alkali, which is at 8.5. As the pH also can change the hydrogen ions of the active sites of the enzymes. And substrate surface. Because of they have not compatible, the enzyme substrate complex cannot be formed. When the pH is not at the optimum level, the reactions cannot be taken place, and then as the pH go back into its optimum pH, and then the enzyme can return as normal. But if there is an extreme pH, it can break the structural chemistry bond inside of this active site and substrate and cause the denaturation of the enzyme. And this is basically what's the figure shows, like the molecule of the normal enzyme that have denaturation process happen. It undergo denaturation and become an enzyme that are break in their structure. The third effects of enzyme reaction is the substrate concentration. In this that kind of effect, it telling us that the substrate concentration is increased and the rate of reaction also increased. Concentration of substrate increase, but concentration of enzyme within the system is fixed. Okay, to the point number one. The point number one, we can say that as the substrate concentration increase, the effective collision also increase. Effective collision between the substrate and enzyme molecules also increase. And then the second point within the first point is it will increase until reach the maximum level. And here is the maximum level. After reach the maximum level, the reaction is constant, become constant. And then the point number two, after it reach maximum level, we can say that the concentration of enzyme that already exists in the system is equal to the limiting factor. It becomes a limiting factor. So we can increase the rate of reaction by increase the quantity of enzyme, the concentration of enzyme. And then after reach the maximum level, all active sites of are saturated with substrate. Meaning that all of the active site of the enzyme had already bind with all of the substrate. So there is no place for substrate to combine with the enzymes. Okay, the last factor that contribute to the enzyme activity is the enzyme concentration. So here, this is a graph showing the effects of enzyme concentration, which is the rate of reaction against the enzyme concentration. So there are 3 points that we need to know. The first point is about when the enzyme concentration is increased, and then the second point is about if we increase the enzyme concentration and at the same time we try to increase the concentration of the substrate. So as long as there is a excess supply of substrate. And then the third point, we try to tell the maximum level. What happened during at maximum level? Okay, so let's we look at it. For the first point, as the enzyme concentration increase, the rate of reaction also increase. Because why? The reason is because there is a presence of the reason is because presence of more active site ready for catalytic functions. Okay, for the second point, we need to have the amount of substrate converted to products per unit of time. If we make a probability, if the concentration is double, converted to products as long as there is an excess supply of substrate. So here we want to get the concentration of enzyme 2 times, so that the amount of substrate converted to products per time, the unit of time is also 2 times. So we need to have an excess supply of substrate. And then the third point, at the maximum rate, the limiting factor come into the place. Limiting factor. This limiting factor is when the concentration of the substrates when the concentration of substrates already saturated, meaning it already bind with all of the enzyme active sites. So if we want to increase the rate of reaction, we need to increase the amount of substrate within the system. Okay, that's all for the factors that affects the enzyme activity. Now we move to the last topics within the subtopic of enzyme, which is application of enzymes in daily life. So as we know, there are a lot of commercial sector for everyday use that needs to use enzymes in order for reactions occur very fast and we can get the products very easily. And these usually are enzymes extracted from natural resources. For example, the bacteria. And some enzymes also produce synthetically. How it produced synthetically? And these enzymes is called immobilized enzymes. Usually these enzymes combine with inert and insoluble substances to increase the resistance of enzyme towards change in factors such as pH and temperature. Thus, immobilized enzyme is an enzymes that are combined with other substances which are insoluble substances to increase the resistance towards change in factors such as pH and temperature. And this technology is called immobilized enzyme technology. Application of enzymes. First, we have protease. This protease can digest the protein into polypeptides and other amino acids. And usually it used to separate the fish skin. And then digestive enzymes, which are pepsin, can also digest our food inside our stomach. And lactase can digest lactose into galactose and glucose. And then pectinase, this pectinase usually found inside juice production. And amylase, this amylase can found inside our mouth, as well as lipase to inside our digestive system, also protease. And the cellulase enzymes can digest cellulose into glucose. And then we have one last application, which is trypsin, that can be found inside our small intestine in the ileum, and it must be in alkali medium. Since we already learned all about enzymes, and now it is time for us to test our understanding. First of all, what is the term of active site? Active site is the region that can be found at the enzyme, and its shape must complement with the substrate. the region of an enzyme to which the substrate binds. The active site has a shape that complements its substrate. Second question. Which of the following enzyme and temperature suitable to completely remove cooking oil from towel? Amylase at 10 degrees Celsius, amylase at 35 degrees Celsius, lipase at 10 degrees Celsius, and lipase at 35 degrees Celsius. First, we need to know the properties of the cooking oil. It is an unsaturated fat and mostly composed of lipid, and this lipid needs use enzyme lipase, and the temperature must be at optimal temperature so that all the reactions can take place. So the answer should be D, because optimal temperature is our own body temperature, which is 37 degrees Celsius. Okay. Diagram below shows an experiment to study the action of pepsin on albumin suspension. Which of the following steps could increase the rate of reaction of pepsin? Pepsin is an enzyme that can be found inside our stomach, and it needs to be an acidic condition. Add few drops of sodium hydroxide solution. This is an alkali medium, so it can lower the pH or increase the pH. This sodium hydroxide solution can increase the pH, so it is not suitable. Increase the temperature of water bath to 37 degrees Celsius. Yes, this is true. Increase the volume of the albumin suspension. Since we look here, the albumin suspension has 10 milliliter while the pepsin is only 1 milliliter. Here we know that pepsin is less so that it can take action very quickly and effectively. Increase the concentration of pepsin to 2%. We can increase the concentration of pepsin so that all these substrate can take place into the active site. Okay, the answer should be 2 and 4. The answer is B, C. Yes, C. And then number 4. Why are apples that have been boiled after they are peeled do not change color to brown? Apple contain a type of enzyme that transforms the color of apple tissue to brown. If apple is boiled, the enzymes becomes denatured, causing the apple to not turn brown. Here, we need to tell that apple has kind of enzyme. This enzyme able to transform the color after it's oxidized to the air. And when we boil the apple, the enzymes destroy, or we call it denatured. And this enzyme, since there are no reaction of the enzyme happen anymore, this apple do not turn brown. Let's we sum up all the topics that we learned inside of this chapter 5. The name of our topic is metabolism and enzymes. And we have learned 3 things. We learned about the metabolism, we learned about the enzyme, and also the application. Metabolism have 2 types of reaction, which is anabolism and catabolism. Anabolism is when we want to make a complex substance from a simple substance, while the catabolism when we want to create a simple molecule from the complex molecule. And then we learn about the energy, which is anabolism. Energy is absorbed, while the catabolism energy is released. And then we learn the nomenclature, which is the addition of the word ASE. And then we learn the general characteristic. We learn about intracellular and extracellular. Lock and key hypothesis. And factors that affect the enzyme's action, which is temperature, pH, substrate concentration, and the enzyme concentration. Now about application, we learn the digestive enzyme, digestive enzymes such as pepsin and trypsin, and then we learn about lactose that we can be found in the milk and fruit juice, biodetergent, yeah, that's all we learn about this chapter.